Gab, utropod. La, FB is life. Are we complete na, ma'am sir? Let's. By the way, kinsay friend din ni Miss Zapanta, ma'am sir. She is not responding to my PMs. Miss Barcosa and I are worried because she was absent for like 2 weeks. So if anybody can contact her, please tell her that I said hi. Good morning. All right. We need to begin our Monday session as always. We should start at 8:30. But anyway, if I can finish earlier, then you will have a bit more leeway moving into the next period. So that's well and good, right? Uh, like I've said, we cannot rest on our laurels despite the massive success of last week's event. We need to continue and push on with our learning. So the lecture will push through as planned, right? Uh, the other thing is that today's lesson is very, very easy in the sense that number one, I have let you done. Is that grammar correct? I've let you accomplish an activity weeks ago about trying to research about different functionalities of the cell, and the other thing is that this lesson is typically introduced in high school biology. So this will be a review, nothing more. That said, I will be moving very very fast with a lecture this morning so that we can finish and move along, right? Because the other stuff that we might talk about are stuff that aren't focused on in high school biology. So let us deal with it before moving on to the more complicated stuff, right? So I'll be turning off my video because I'm not important. And then we'll proceed with my screen. So your confirmation if I am correctly sharing my screen. Let me see if I can do that. All right, I'm not yet sharing my screen. Damn it. There we go. Do you see the screen on your end?
Yes,
It
sir.
should
Yes,
read
sir.
Modiel
Yes,
2
sir.
cells, correct. Before that, allow me to personally thank and congratulate you for all of your efforts in last week's science and mathematics convention. I'm very, very impressed, Mom Sir. You deserve to be thanked and congratulated in person, but I cannot do so right now, so this will suffice, right? Now, I and my teachers have agreed to reward you with the following components. The sorry, the accomplishment of the ticket, meaning you have all four stamps. Asa na to? There we go. You have all four stamps accomplished will constitute one written work in my subject as well as in physics and chemistry. Now, a written work is typically weighted at 30 points, so that will be 90 points total, free of charge for as long as you've completed the ticket, right? The accomplishment of your exhibit will be two performance tasks in my subject as well as in physics and chem. So that will amount to 300 points total. Okay? We have been thoroughly impressed when we talked about things last week. So we will also waive the first quarterly examination in my subject as well as in physics and chem. There will be no written component here. Instead, we will be featuring an alternate activity. The physics teachers have already told me that they are planning to stage a moving exam, and in our case, Ms. Cosido and myself have decided that your microscopy practical test will be your substitute for the written first quarterly exam. So this will be the package deal for all of your efforts, which we were very, very grateful for during last week's event, right? So congratulations to the team, Maricela and company, no, for winning first runner-up in the competition last Thursday. So congratulations, everyone. I cannot thank you personally because all of you did your part. So once again, thank you very much and congratulations. Okay, so napay ni Sud. So let's admit. However, for those who did not greet me this morning, you will be given an essay type quarterly exam because you have offended me. Okay, wa ni greet. So ato i-recall. Kinsay ni greet everyone from Holly to John. The rest, Robert ni Apas 8:30. Anything beyond Robert o Aris, ala sige. Sige gyud. Paningkamot. Naningkamot yung nahadlok sa essay. Anyway, so that's the package deal and that's for all of your major subjects. So it's a job well done and you deserve that because of the massive effort that it took to accomplish last week. We couldn't have done it without you. Okay, finally, that aside, we will move on to today's discussion. We will finally begin with our discussion of cells. And like I've said, this is typically reviewed, right? Because this has been taken up before. So this bell work is immaterial. This is here for one purpose only. I find it difficult sometimes to conceptualize biological constructs, right? We cannot see the cell outright, so it's sometimes difficult to imagine what they do. In my case, I found it useful to compare analogies to cells, right? Analogy. I find it useful to think of analogies in order to help me better understand the cell. For example, instead of taking a look at the cell, I take a look at a city or an organization because the cell, being self contained, functions similarly to how a city functions, right? And in order to understand better what organelles do, I sometimes compare them with parts in a city, such as the government, the power sector, the industrial sector, your factories, and the like. This allows me to understand the cell on a more human level. For those of you who are fond of anime, you may be interested in an anime known as Cells at Work, which is available on Netflix, by the way. And this one also provides a different analogy for cells. They try to represent cells with anthropomorphic characters, right? Similar to human beings that happen to be found in your body. So the main characters of that anime is the red blood cell and the white blood cell, and I found it quite helpful for those of you who are inclined with that particular work of art, right? But in our case, this will occur very, very quickly, like I've mentioned. On the left side of the screen, you are seeing a representation of onion cells. So I have shown you this via the microscope. If you are familiar, if you're rather, if you remember the microscope slide that bared the onion slide or even the lily slide, it will feature a rectangular or geometric appearance of the plant cells, right? They typically occur in that fashion. Although in our microscope slides, the onion cells were dyed pink with saffron in this case, they're actually colorless because if they're only one cell big, we often cannot appreciate the colors and pigments in the cell. I've mentioned before that cells are the smallest unit of life as well as organization. That is because cells possess the characteristics that are present in living organisms. Furthermore, cells are self contained. They are independent. They can function all by themselves and continue doing so, although normally they function alongside their other cells in order to contribute meaningfully to the organism. True for all cells is that they are covered by a protective covering called the cell membrane, which serves a lot of functions. Number one, it serves as an additional layer of protection for the cell. And number two, it provides a boundary. What is inside the membrane is considered part of the cell. We refer to that as the intracellular space. What is outside the membrane is known as the extracellular space, and that is beyond the confines of the cell. The cell membrane serves a protective function because it is semi permeable, which means that it allows some substances to cross the membrane, but it blocks the entry and exit of other substances, especially those that are important for cellular function. Inside the cell membrane, we can find something that is gelatin like. If you have ever cooked with eggs before, it will be similar in constitution to the egg white, right? It's a bit slimy and it's filmy and watery because that structure, called the cytoplasm, is composed primarily of water with other inclusions and substances mixed alongside it, right? This gelatin like substance comprises the contents of the cell. Scattered throughout that substance, which we call the cytoplasm or the cytosol, are the other cellular parts, which are called organelles because they are tiny organs, right? Now, depending on the book that you're reading, the cytoplasm can be also the cytosol. It's the same thing. The cell membrane is often called the cellular membrane or the plasma membrane, right? The specific branch of biology that deals with cells is called cytology. When it comes to tissues, it's called histology. Those of you who are looking at a career in med tech, histology will form a large portion of your diet, right? Now, cells are quite important because the similarities and differences among the cells in living organisms have allowed scientists to divide them into different classifications. Under domain Eukarya, you find all of the common and familiar organisms, primarily plants and animals, right? Fungi are quite different. Their cells resemble animal cells, but morphologically speaking, the way they look like, it's a bit close to plants, although distinctively, they cannot photosynthesize. So they have a different method of nutrition compared to plants. What about kingdom Protista? Simply put, if you are weird, you are lumped by scientists under this kingdom. Under this kingdom, you will find the algae and the protozoa, which comprise plant like, animal, and fungus like organisms, but aren't animals, plants, or fungi, right? A good example is Euglena viridis, which looks like this. It has a flagellum with which it uses to swim through water. It can move about like an animal. Much like an animal, Euglena viridis also has a mouth part, an opening which allows it to eat smaller organisms, such as molds, other fungi, and even bacteria. So it eats much like animals. It is only one cell big, so it does not have the organ systems that are often thought of when we think of animals. But why isn't it considered an animal? Again, it's only one cell big. The other thing of note is that in the absence of food, Euglena actually can photosynthesize, meaning all it needs is a bit of light and it will trigger organelles in its body to manufacture glucose. It can produce its own food, much like a plant, but it can also eat stuff, much like an animal. So if you are weird like that, scientists lump you in kingdom Protista. They aren't sure unsa man gyud ka, so gimugnaan sila kingdom all by themselves, right? The other domains have different cellular orientations. Domain bacteria and domain archaea are prokaryotic in nature, which we will get to in a while. But let's begin with what it means to be part of domain eukarya. Simply put, domain eukarya contain eukaryotic cells, which roughly translates from Greek as true nucleus. Eukaryotic cells are large and generally more complex. They have a well defined nucleus, a centrally located organelle which serves as the control center of the cell. They also have complex organelles which are bound by membranes of their own, which is similar in structure to the cell membrane surrounding the cell. All organisms that are animals, plants, fungi, or protists have eukaryotic cells, and that is why they are grouped and lumped together in domain eukarya. The other two domains feature prokaryotic cells, which roughly translates to before the nucleus. They are quite simple, right? They're a bit ancient. They were the first life forms that originated in the planet. They do not have a nucleus compared to the other cell types, nor do they have any of the organelles except perhaps the ribosome. This is the only thing that they possess. They do not have the other parts of the cell found in eukaryotic cells. The simpler organisms, the single celled bacteria, and archaeans all have prokaryotic cells, which will now lead into the discussion of what are the different parts of a eukaryotic cell. So, this is in large a review of what you've talked about in high school biology. Like I've mentioned, the biggest part of the cell is its nucleus, which is often centrally located and darker stained compared to the others. For those of you who have looked at the paramecium slide, which I provided in the sketch and scope activity, oftentimes ang inyo gidrawing ingon ani ra guys, no? Which is correct. It's not wrong, but you often missed out the darker stained part inside the paramecium, right? Which is actually the nucleus of the cell. So it can be easily seen. In fact, our microscope slides emphasize that, but few people actually picked up on it, right? The nucleus is quite important. It is bounded by its own membrane, termed the nuclear membrane, and it is a darker inner area known as the nucleolus, which constructs the ribosome, which is the second time I've mentioned this today. right? The purpose of the nucleus is to direct cellular functions. It will tell the cell what to do and how to do it because it contains a very important molecule, the DNA, which comprises your genome. DNA contains genetic information. We will talk all about DNA once we reach module 2. For now, suffice it to say that DNA is the blueprint of the cell structure. This is what the instructions for building the cell, right? This is also a manual for how the cell functions. Basically, DNA instructs the cell what to build and how to do stuff. And it's located in the nucleus. And for that reason, the nucleus directs cellular function, right? The other organisms, no, no, sorry, the other organelles, the mitochondria and plastids, are lumped together here because they serve in metabolic functions. They give the cell energy. Mitochondria, the sausage shaped organelles you find there with lots of folds inside where cellular respiration takes place, are found in all eukaryotic cells. Plastids are only present in photosynthetic cells, plant and algal cells, those organisms that are capable of photosynthesizing. Plastids are, let's discuss it first, right? Plastids contain pigments necessary for photosynthesis. The chloroplast, pictured here, contains the pigment chlorophyll, which gets excited whenever it captures light energy, right? Which triggers reactions that allow it to manufacture glucose, or chemically C6H12O6, right? Now, the glucose is what cellular food is really. Grammar is wrong. Glucose is what cellular food looks like, something to that effect, right? What is this glucose used for? Glucose, if it's a plant, it will be given off to the mitochondria. In animals and fungi, it is typically obtained in another way. We need to eat, for example, to get glucose, right? We get that from Go Foods. Glucose, regardless of where it was sourced from, are used up by the mitochondria in cellular respiration. Glucose, alongside oxygen, reacts to form carbon dioxide, water, and a very important compound, adenosine triphosphate, which is cellular energy. Whenever ATP is broken down, it releases energy, which powers a lot of the cellular activity. So, simply put, the mitochondria is where the magic happens. Inside it, cellular respiration takes place to supply the cell with ATP, which the cell in turn uses for various other functions: governing daily activity, reproducing, building other cells, building cellular products, right? This happens in the mitochondrion, which is why kada pangutanon ang biology student si nagkatunan sa biology ang ilang tubag basta the mitochondrion is the powerhouse of the cell. Mora na ilang matubag, no? Very, very meme worthy. There you go. Another important part is the layer of membranes, which are collectively called the endoplasmic reticulum, or often shortened to ER. Now, it is nothing more but a stack of membranes, which is similar to the next organelle, which is also a stack of membranes. So, how do we tell the endoplasmic reticulum and the Golgi apart? The endoplasmic reticulum is adjacent to the nucleus, it is found side by side the nucleus. From the nucleus, the instructions to build stuff then goes into the endoplasmic reticulum, which serves as the factory of the cell, which builds various cellular products, right? Now, depending on the type of endoplasmic reticulum, the cell builds different things. Part of the endoplasmic reticulum contains the dot like structures you see on screen, which are referred to as ribosomes. Each ribosome manufactures proteins. So the part of the endoplasmic reticulum with the ribosomes builds proteins. Because on microscopic examination they look rough, It is called the rough endoplasmic reticulum because of the abundance of ribosomes. The other part of the endoplasmic reticulum without ribosomes looks smooth, and it's therefore called the smooth endoplasmic reticulum. It will synthesize non-protein products such as lipids, for example. Okay? So with whatever the cell needs, be it proteins or other substances, parts of the endoplasmic reticulum get activated to produce those. Once those have been produced, they are then sent to another layer of membranes known as the Golgi apparatus, the Golgi body, or sometimes more recently we just call it the Golgi because why not? Again, the Golgi is situated farther out from the nucleus compared to the endoplasmic reticulum, which is adjacent to the nucleus at all times, right? Now, the Golgi apparatus does not manufacture products. Instead, it receives products from the endoplasmic reticulum, stores them, processes them. Sometimes it matures them so that they are usable, and then it packages them into containers called vesicles, which are temporary membrane bound spaces which contain a variety of things. Cellular products built by the cell. From here, that vesicle will pinch off from the Golgi and it will move to other parts of the cell to distribute those products wherever they are needed. So not only is the Golgi a receptive organelle, it will also distribute it around. So the cell has its own delivery app, right? It uses the Golgi and the vesicles for that. Like I've mentioned, the vesicles are the tools with which the cell transports various chemicals and substances throughout the cell as well as outside of it. It is also what cells use to receive stuff from outside down to the cell. Vesicles are nothing more than temporary, membrane bound spaces that are used for containment, storage, and transport. A category of vesicles termed lysosomes do contain something special. They bear with them digestive enzymes, which the cell uses to dissolve stuff. These stuff include waste matter, worn out or damaged or infected organelles, and stuff that find itself inside the cell. For instance, if a bacterium manages to find its way inside of the cell protects itself by trying to fuse that bacterium with a lysosome so that it gets destroyed, right? Think of these as mobile disposal units of the cell, also useful for recycling and cleanup, right? Finally, we reach the last organelle, not organelle, but rather a component of the cell, right? We call that the cytoskeleton, a series of fibers that extend the entire width and breadth of the cell, which helps it to hold its shape. Now, this is critical. Even if we call it the cytoskeleton, ladies and gents, this isn't fixed like our skeleton. The cytoskeleton can be disassembled and rebuilt in order for the cell to accommodate changes in shape. So if it needs to squeeze, right? Or momentarily murag muhiyak ang cell, ma'am sir ba? The cytoskeleton shifts and changes to allow the cell to change its shape. So it isn't hard fixed similar to our skeleton. It's a bit more fluid and dynamic, right? It can be disassembled and built again. All right? The cytoskeleton serves as the infrastructural support of the cell, holding the shape. Furthermore, it also allows the cell to compose locomotory organelles if in case it needs to move. So these are organelles used for motion, right? There are four in particular. The first one are short hair-like extensions that look like eyelashes. We call these the cilia, which are present in organisms like paramecium, uh, featured here. They wave about in a uniform motion to propel the cell along. This is how paramecium swims. The other one is a long whip like organelle called the flagella, which it uses to swim through fluid. So, a very good example is the sperm cell of human beings, but pictured here is the aforementioned Euglena viridis. Both of these cells are flagellated. The single flagellum allows them to swim through water or whatever's found inside the vagina, right? Vaginal fluid? I'm not sure. Pseudopodia are yet another organelle, but they're temporary. They are extensions of the cell membrane, which a cell uses to creep about a surface. Pictured here is amoeba proteus. It uses pseudopodia to move as well as capture other organisms for eating. We will discuss about how pseudopodia feature in one type of cell transport. The other rare organelle are the myonemes, which are an extendable stalk which can retract and coil, thereby pulling the organism down. Pictured here is the protist vorticella, which has a myoneme, right? I think that's Vorticella. I could be wrong. You might want to check on that. Like I've said, no, I might lie in class. But generally speaking, that's how cells work. Now, in the human being, I have mentioned that sperm cells contain flagella. The cells lining the respiratory tract also have cilia, but they're not used to move. Rather, they're used to create a wave like motion so that the respiratory tract can propel mucus upward so that it reaches your throat, whereupon you can swallow it or spit it out, right? It traps mucus and debris. And the mucus, rather, traps debris and other stuff that finds its way down to your respiratory tract. I bet that's it, right? Now, let's pause for a while and get this learning checkpoint. Instead of asking you a question, I want you to try and fill out the following. Let's consider that number one, number two, and number three. Let's tackle number one first. Allow me to read. All blank cells possess a nucleus and membrane-bound organelles. This makes them distinct from blank cells, which do not have nuclei and other organelles. These differences in cell structure led scientists to classify these organisms in different blank. So can you tell me what the answers are to these blanks? You might want to write your answers in chat. Just write the three words one after another, and then I'll reveal the correct answer so that we can ascertain whether we are on the same page or not. Robert, very, very fast. Thank you very much, sir. Let's go. All eukaryotic cells possess a nucleus, correct. This makes them distinct from prokaryotic cells. And this is the reason why scientists classify them in different taxa, kingdoms, domains. Pick your answer. All right. So kingdoms are correct. Taxa is also a correct answer. Classification grouping. Domain is also correct, as is kingdom. If you go cheesy, you can say different groups. That will also make sense, right? Let's tackle number two. Thank you, Robert. The rough endoplasmic reticulum is the organelle in charge of blank synthesis. This is due to the presence of tiny organelles called blank. Cellular products made from the endomembrane system are then sent to the blank for storage and distribution. The endomembrane system is the collective term for the endoplasmic reticulum because it is a membrane system inside the cell, and it's a very wide network of membranes, which is why it's called a system. Do I have answers? No, I do not. Nag-FB pa sila. Let's get the final answers. The rough endoplasmic reticulum is the organelle in charge of protein synthesis, right? This is due to the presence of tiny organelles called ribosomes. Again, these are the dot-like structures seen in the rough endoplasmic reticulum, right? Each of these is a protein factory. We will talk about them in greater detail once we reach Module 2. Cellular products made from the ER are then sent to the Golgi for storage and distribution. Let's tackle number 3. The blank in the cells of plants enable them to photosynthesize. One example is the chloroplast, which contains blank. Glucose, made from photosynthesis, is then used up by the mitochondria to produce molecules of blank, which cells use for energy. I'll give you a moment to think about that. It's getting warm. It rained a bit this morning and then now it's getting warm. What the hell, weather? All right. Do I have any answers for number three? Robert, correctly to ganina silang Margaret o Vera. I'm so sorry I didn't quite catch that, right? So we have the plastids in the cells of plants enable them to photosynthesize. One example is the chloroplast, which contains chlorophyll. Now, please take note, do not limit yourself to just chlorophyll. It isn't the only photosynthetic pigment. Others include xanthophylls, right, which give the plants different color. You have phycobilins, which give it a red or black phycobilin, red or black coloration. You have carotenoids, which grant it the yellow, orange color. Different pigments, but the most common, the one that's green, and for which reason plants generally look green, is because of the abundance of chlorophyll. Glucose made from photosynthesis is then used up by the mitochondria. Take note, mitochondria plural, mitochondrion singular, to produce molecules of adenosine triphosphate, which cells use for energy. All cells utilize ATP. They just produce it differently, right? Some go for cellular respiration, others do it by fermentation, others get glucose from chemosynthesis and then utilize a different mechanism for breaking down ATP or creating and then breaking down ATP. Correct, Lois, you got it right, ma'am. Let's go. So to finish the analogy, which is immaterial as of the moment, I think of it like this. No? The nucleus is like the government. Mo may magbuot. Siya po'y murag educational sector because it keeps a record of genetic information, the DNA, right? The power sector is obviously your plastids, which produce the raw material, and mitochondria, which make use of the raw material as fuel for cellular processes and activity. Right? Again, mitochondria are in charge of, excuse me, cellular respiration. The industry of the cell is governed by the endoplasmic reticulum, which builds and manufactures cellular products. Right? Those are then received god received by the Golgi body, which serves as the commercial sector. It also takes charge of distribution via units called vesicles. And if the cell needs to clean or destroy worn out organelles or debris, it uses lysosomes for that purpose. And finally, to hold the cell structure in place, you have the cytoskeleton, right? Any questions so far while I drink water?
None, sir.
Why question, ma'am, sir? Ka-cute ninyo. Eh, what may question ng mga tao? Okay, I will proceed if there is no question, right? The following, my voice is, God, so haggard. The following organelles pictured in maybe these next three slides aren't found in all cells, but specialized cells will feature them. For instance, in animal cells, you find a pair of organelles called the centrioles, which are located in a space called the centrosome. Centrioles are similar in structure to the cytoskeleton, but they have a very unique purpose. The cells use them during cell division. We will talk about that once we reach cell reproduction farther down the line, right? You only see them in animal cells because plant cells, when they undergo mitosis, follow a little different approach, right? It's a bit um, dissimilar from how animals do it. So, for that reason, some of the cells do not need centrioles to maintain the cell shape as mitosis is happening, right? Other cells will also feature an additional layer of protection. The cell wall is a protective layer in addition to the plasma membrane, so they are not the same. Huh? All cells still have plasma membranes, but not all will have a cell wall, right? The cell wall is found in the following organisms, but it's made up of different things. In plants and algae, it's made up of a carbohydrate called cellulose. In fungi, it's made up of a protein called chitin, which is strangely the same substance making up the shells of crustaceans, shrimps, crabs, lobsters, and also the exoskeleton of insects, right? So if you're familiar with beetles like the rhinoceros beetle, kanang bakokang, ma'am sir, the hard plate-like covering of that insect is made up of chitin, which is the same material making up the cell walls of your mushrooms and your molds, right? Bacteria have a very complex cell wall composed of a glycoprotein. Peptidoglycan is both a carbohydrate and a protein. This is so that there is an additional layer of protection. Okay, bacteria are quite small and simple. They don't have massively complicated defenses. Instead, they rely on their cell wall to survive, right? In fact, antibiotics are targeted at destroying the capacity of a bacterium to produce its cell wall. So if we get rid of the cell wall of a bacterium, it will then be vulnerable to the other defenses of the body, right? Archaea are unique. They are like bacteria in all regards, except that their DNA is different and their cell walls are composed of weird substances. Others are pseudopeptidoglycan. Others have not yet been determined by scientists. It is very hard to study archaea because they typically live in extremely hostile environments, which means that laboratory setups need to mimic that environment in order for them to thrive. So some archaeans live in extremely salty, extremely high pressure, extremely hot, extremely radioactive environments, which means it's hard to simulate that in a lab environment. So studying them is complicated. Nonetheless, they are amongst the first life forms to have appeared on the planet when the Earth was still relatively hostile compared to now. So studying them, like I've mentioned, is a bit more difficult. Now, cellulose is why those cells are quite geometric in appearance. So if you recall the onion and lily cells that I've shown in the lab, it will look square-like in appearance, right? Because of the cell wall. You can very easily see here that there is a membrane and another outer layer. So this is the plasma membrane of the cell. This is the cell wall, outer covering. It's also green because of the chloroplasts, and also quite obvious is the nucleus of the cell, right? In this image, you can only see the cell wall as well as the chloroplasts, right? The cellulose in plant cells is what gives it the distinct crunch. So if you're fond of eating cabbage, carrot, onion, if you bite into those things, there is an audible crunch. That's because of the cellulose in those cells. Okay. Finally, plants also have a unique vacuole. Now, do not be confused. A vacuole is just a glorified vesicle. It's a bit bigger. It's a bit permanent. It's Compared to a vesicle, it stays in place. In fact, plants have a large vacuole in the middle, which contains substances, so it's useful for storage. It also controls the pressure in a plant cell, and it serves detoxification purposes. The reason for this is that lysosomes aren't found in plant cells, so the function has been taken over ah, by their vacuole. There you go. So I have here a summary what plant cells have and what animal cells have. To distinguish between the two, you have a cell wall. Animal cells do not. Plant cells have plastids and mitochondria. Animal cells only have mitochondria. They cannot photosynthesize. Plant cells have a large central vacuole. Animal cells use lysosomes for cleanup duty. Plant cells do not have centrioles. They do not need that for cell division, nor do they have a lysosome. Animal cells do. This is by and large the distinction between the two. Take note that the plastids comprise your chloroplast, which gives plants their telltale green color, right? Now, where do we go next? I would not like to discuss this because reasons. Let's see if you can figure this out on your own in your asynchronous period. I want you to walk through the three remaining slides I have and discover for yourselves how prokaryotic cells differ from eukaryotic cells. Again, these are the cells of your bacteria and your archaea, the two other archaea. There we go. So Let's see why they are different and how they are different. So I have in this slide the fact that they do not have a nucleus. Why? Read this bullet. The other is that they do not have organelles. Why? Read this bullet. And the other is the other stuff that bacterial cells possess. So we have fimbriae, pili, and plasmids. So let's take a look at what each of them do. I'll let you read this all by yourself for your asynchronous days, and I'll pick this apart next week. And also other protective features of bacteria, as well as the shapes that they possess. And once you're done, I have here a short summary highlighting the differences between the two. And finally, a final question. Which features are present in all cells? So, after reading through the remaining portion of the discussion, I want you to try your hand at answering this question. Any questions for the meantime? What
None,
I say
sir.
questions? You always never ask me a question. What if I do not dismiss you? No dismissal yet.
Excuse me, sir.
Yes, go ahead, Kirill.
Dili siya sa lesson, sir, ba? Ba't kato ganing activity katong mold
Yes.
something?
Uh-huh.
Nagkuhan pa tato ikaw Wednesday, sir.
Yes, ma'am. It will push through. Ngano
Okay,
deg
sir.
Wednesday, nag-prepare mo og something.
Hello, sir. Sikreto, char.
Expectant kayo. We will push through. It's a very short activity. It will need maybe 10 minutes of your time, and that's it. Yeah. Take note that it's individual, so it's by group, but the accomplishment of the worksheet will be individual. You will need to draw your own. So either print a copy of the activity guide on the LMS so that you have a guide on what to do, what steps to take, and then I've also provided an area for you to sketch your drawing. But if not, if you don't have a printer, perfectly fine. Just bring paper along with you. Have the guide on standby in your device so that you can read the steps and then just draw it in another piece of paper. I'm not quite strict, but it will be another part of your portfolio. So it will have two contents right now, the scope and sketch activity, and this one. Okay. And we will also add another one next week once we accomplish the first experiment. So I hope I've clarified things. Anything else? So that's on Wednesday. Please take note of the things you need, the mold, and the personal protective equipment. Yeah? Anything
Yes, sir.
else?
Thank you, sir.
Thank you,
Bye.
Kirill. Steve says hi. Hello. No questions, 1A. May bang discuss na ko prokaryotic de eh. Ayaw dito ra kutob kay para challenging po ang inyong experience pa kay what jud ko ihatag asynchronous reading ra gyud. So let's keep it at that. If there are no questions, ladies and gents, thank you very much for attending our session this morning. You have a great day ahead. Bye, guys. See you around. Have fun. See you.
Bye, sir. Thank you,
See
sir.
you in
Bye.
chat
Thank
Bye.
you,
Thank you,
sir.
sir.
Goodbye,
Bye,
sir. Thank
sir.
you.
for
Bye,
whatever.
sir. Bye, sir.